Wow, a lot of people. Good afternoon. My name's Stephen George. I'm the editor of Discover Magazine, and I wanted to welcome you all to our event, Programming Life, the Revolutionary Potential of Synthetic Biology. And we have a lot to get through this afternoon, and I know nobody came to hear me talk, so I just want to thank SynBERC for hosting this event, and thank the panelists and our moderator for taking time out of their busy schedules to attend, and thank you all for coming. And so without further ado, I wanted to introduce the director of SynBERC, Jay Keasling.
Thanks very much. I too want to welcome you here to our session on Programming Life. Our discussions today will range from the challenges of finding the right tools to build game-changing efficiencies into our work to the applications in use today. We'll also consider what's ahead for our field, the potential for synthetic biology to help to move America into a true bioeconomy, and we'll talk about the concerns that all of us share about the social and ethical implications of our work. We hope you enjoy these discussions today and continue to engage with our field online on Discover's website, where you'll find additional resources for synthetic biology. I'd like to take the time now to thank Discover Magazine for helping us to put this together. In particular, I'd like to thank Steve George, Cory Powell, Darlene Cavalier, and Brianna Draxler. We appreciate the opportunity to talk to your readers about the emerging field of synthetic biology. And now I have the great pleasure of introducing our keynote speaker, Juan Enriquez. You may know him through his TED Talks, where he asks provocative questions such as, will our kids be different species? Mine obviously are. And if you'd been home with me this morning, you'd know that. The Discover channel calls him a curiosity expert, and he is well known for his discussions of how the life sciences will drive profound changes in our core religious, government, and social structures, but most of all, in all of us. Juan is a managing director in Excel Venture Management, a venture capital firm that invests in life sciences technologies. He was the founding director of Harvard Business School's Life Sciences Project. He also co-founded Synthetic Genomics, a synthetic biology company focused on developing and commercializing genomic-driven solutions to address global energy, medicine, clean water, and food challenges. Please join me in giving a round warm of applause for Juan Enriquez.
So I'm here today primarily as a map maker, because I've got all the discoverers in front of me. And it's a sign of where we are that you can put most of a field in the front part of an auditorium. And if you brought a few more people, then you'd have most of the field and most of the discoverers. And what you're going to see today, and it's a privilege to see it up close, is a little bit of an atlas of where we're beginning to discover the edges of known worlds. And this is difficult. You're going to hear some stuff today that is highly technical, highly nerdy, in a different language. And the thing which I want to explain a little bit is why it's worth learning this language. Because it's difficult learning a new language. I mean, if you're going to go out and take on Chinese, or if you're going to learn new French, you know, you found a boyfriend or a girlfriend, or you're moving, or you got fascinated by the literature of a country, but you've got to put an enormous effort into learning a new language. And of course, humans keep doing that, because humans are the only people who use language. And because we use language, we're able to teach the next generation and the one thereafter and the one thereafter what we were doing when we were using hieroglyphs in a French cave 2,000 or 3,000 years ago. And that was enough to transmit knowledge of how you hunt and how you do fire and how you do this and how you do that. But of course, it wasn't very effective because you couldn't transmit that data to a lot of people. So we invented a new language, and we put it on papyrus, we put it on paper, and that was a transition between tribe and empire. And the reason for that transition is all of a sudden information could move across very broad distances. So you could transmit discoveries, you could transmit what you were learning, you could transmit what you were doing, what you were beginning to execute on, and the great tribes, the great empires, became those able to adopt and adapt to new discoveries. That, of course, became ABCs. We had movable type, and over the last 30 years, we transitioned into ones and zeros. And if I'd stood in this auditorium in the late 1970s and told you the single most important language in the world, the language that is going to be 99% of all data transmitted today, is ones and zeros, you would have looked at me and said, this guy is crazy. And the interesting thing is, we're going through that transition today, and what you're going to hear during this is part of that transition and how that transition is evolving. So think of these folks as explorers who are coming in to give reports of the edges of the known world, and they're taking you into what's new this week, this month, and what they'll find over the next year, and how this stuff will change your life. And again, it sounds obscure, it sounds distant from what you're doing, and just as in the late 1970s, we couldn't have imagined some of the very large companies and some of the uses for digital code and the upheavals led by digital code, it is difficult to imagine the transitions led by synthetic biology. And as you're thinking about this structure, first, it's a paradise for academics. And it's a paradise first because you can be an explorer, you can discover new fields. And if you just think of the new fields discovered over the past decade or 2 decades, you start out with genomics and this little obscure reading of the 4 base pairs of DNA becomes an enormous explosion of research and discovery and the human genome, and then the genomes of plants and the genomes of microbes and the genomes of this, that, and the other. Enormous departments at universities. But of course, that's just getting started, because that's a bounded 3.2 billion letter, 2-dimensional puzzle in 4 letters. By the time you get to proteins, then you're talking about 20 some odd little blocks, and you're adding synthetic blocks to that. And it really matters how they fold. So all of a sudden, the computational problems in this become really interesting. And oh, by the way, because that's what executes instructions, you also start getting an impact on medicine, on energy, on textiles, on chemicals, food, feed, fiber. And then you start looking at metabolomics and how does this whole structure operate within the body? How does it all come together? And it gets even more complicated. And then you begin to take things like metagenomics, and you start looking at how entire environments interact, which begins to change fields like medicine in a fundamental way, because each of us has about 100 times more non-human cells in our bodies than we have human cells. And these, of course, are microbes that we have been interacting with for millennia. And as you begin to stack these things, the data volume becomes such that we're accumulating data today about 50% faster than Moore's Law, which means we cannot build computers to process, transmit, order, triage, understand this data fast enough. And that's why you begin to see transitions not just in the biology departments, but you get to get this overlap with computing. You get to get this overlap with information, with data transmission, with Google, with Intel, with IBM, with Microsoft, with a whole series of companies that were outside of the life science field and that now come into the life science field. So what? Well, just as in the last 30 years, a series of startups have become key to the US economy, to the point where 0.2% of the US economy is invested through life sciences and IT venture capital. And that accounts for 11% of US jobs and 21% of US economic output. And just as the companies that come out of Stanford have generated a market cap equivalent to about the 10th largest economy in the world, and a similar study at MIT showed it was the 13th largest economy in the world, although that hasn't been updated. You have these clusters of innovation at Berkeley, at UCSF, at UCSD, in Michigan, in Austin, in Maryland, in Boston, that becomes motors for the US economy, that become the source of most new jobs, that becomes a source not just of biolabs, but the stuff that feeds restaurants and theater and art and libraries and sports and schools and museums. And it's these clusters of innovation that are able to take the work of the new cartographers, the new explorers, and put it to work so that you can generate new things, so that you can build new things, that makes the rise and fall of various regions and states. And as you're thinking about what you're going to hear here today, this is a pretty extraordinary lineup. I'm not going to mention everybody, I'm just going to give you a couple of examples of some of the stuff that you're seeing. And of course, George Church, an extraordinary human being who is always on the edge of whatever is being done. So last week it was de-extinction. Can you take DNA of things like passenger pigeons and bring them back to life? Or mammoths, or dodo birds, or Tasmanian tigers? Working with Stuart Brand, another one of the local, or my local heroes, who started the Whole Earth Catalog and part of the Green Movement. And then you've got Drew Endy, who's let loose thousands of kids throughout the world by founding iGEM and teaching kids how to program in this language of life. And you've got Chris Voight, studying at synthetic biology and mentoring the next generations at MIT. And thinking about some pretty extraordinary things that you'll see pretty darn soon. You've got a really interesting intersection with Densmore, right? So here's somebody who studies electrical engineering, gets a PhD in electrical engineering, is fascinated by video games, thinks he's going to design video games, but actually ends up designing biological circuits. And that intersection between the digital and the biological is going to become more and more important as this becomes a programming language and not just a language of biology. And Smolke, of course, trying to figure out DNA and RNA expression, and how do you begin to express this code, and how do you begin to structure this code? And of course, our host, Jay Keasling, who in his spare time goes out and tries to grow stuff that will cure malaria, and biofuels, and the next generation of clean tech, all the while mentoring students at this university, at the national labs, and creating whole new fields of science. And when you put a group together in a room like this and they begin to tell you about their new language, you don't always understand the words, but it's really important you understand what these folks are doing. And the reason it's important is because this isn't just a transition in new companies, in IPOs, in small companies. This is a transition in some very large companies, which is part of the reason why some very large companies are a part of this debate. When you see 42% of the earnings of DuPont come out of life sciences, that's telling you there's a big transition. When you see 14% of the earnings of General Electric come out of healthcare and life sciences, and that becomes one of the largest and fastest growing areas at a Fortune 50, that's telling you that this stuff is changing. When you begin to see the programs that IBM has using Watson and putting that into healthcare, when you begin to see the amount of data being operated on Intel chips to process life science data, that's telling you there's a big transition going on. And obviously, because you're here, because you're watching this, because you're interested in this, because you're learning this new language, you're way ahead of the rest of the world on this. And it's important you stay ahead of the rest of the world, because not only does it make a difference to your job, to what your kids study, to where you live, these are also important debates because they determine who adapts and who adopts. It determines which countries rise and fall, and you saw that over the last 30 years with ones and zeros. Because no one would have predicted, at least when I was in college, that India would be the second largest software producer on the planet, that Bangalore would be a rising power, that Silicon Valley would be more important than Boston. Countries and regions rise and fall, and they do so on the basis of where these explorers choose to live, where they choose to make their discoveries, where they choose to create their labs. You can move very large economies if you don't make a mistake. The richest state in the United States should be New Mexico, but for the fact that a bank officer didn't make a $500,000 loan to finance the receivables of a little startup called Microsoft. So Microsoft went home and started that cluster in Seattle, along with Amazon and Red Hat and the rest of these things. And it's the places in the United States and the world that attract people like the people who you're going to hear today that are going to be the powers in the future. Because it's brains that power the future. And the last thing I'll say is, this isn't just about economic growth in countries, this is also about where we're going as a human species. Because in the measure that we don't just adapt and adopt to our environment, in the measure that we begin to understand how life is written, how life is coded, how life is copied, and how you can rewrite life, leading to areas like synthetic biology, then you begin to directly and deliberately understand and guide the evolution of microbes, of bacteria, of plants, of animals, and even of ourselves. And as we begin to take direct and deliberate control over the control of evolution, I think we are actually transitioning from a Homo sapiens into a Homo evolutus. That is a creature that begins to directly and deliberately engineer evolution to its own design. And that is probably the most interesting and fundamental cartographic transition going on, and perhaps the most important trend going on the planet today. Thank you.
So I'm Cory Powell. I will be your moderator today. I'm very excited to have all of you here and to have this distinguished panel here. Let me tell you a little bit about the format of what we're doing today. First of all, I want to thank Juan Enriquez for that beautiful introduction. In a sense, what I'm going to do right now is rewind. You've heard a great vision of the potential of what we're doing. And now the point of this panel is to really understand how to make it happen. What are the challenges? What are the techniques? What is synthetic biology and what can it do? And you're lucky that you have the world's leaders in that field right here to give you the authoritative answers. We're going to have 3 panels today covering a lot of material, and I'm going to do my very best to make sure that they're speaking directly to you and directly to the kinds of questions and concerns that you may have. There will be 3 brief opportunities for question and answers. There are 2 microphones on either side of the room, and so we'll have a pause at the end of each session. You can line up on either side and have a chance to talk to the panels. There will also be a little bit of time to talk to the panelists afterward. But I'd like to call up the first panel right now. Now, if you have your program, you have the full biographies of them, and when you're dealing with people like this, I can pretty much spend the entire panel just reading their biographies. I'll skip that and go to the top line. We have on the far end we have Christopher Voight, who is the associate professor of biological engineering at MIT. Then we have Douglas Densmore, assistant professor of electrical and computer engineering at Boston University. And immediately to my right, Christina Smolke, associate professor of bioengineering at Stanford University. Now, I mean, 2 things that I immediately noticed with this group. First of all, they're kind of conspicuously young. And the second thing I noticed with this group is that if you look at their titles, all 3 of them have hybrid titles. These are not, if you want to know how interdisciplinary thinking works, an electrical and computer engineer who is, talking about biology. 2 people whose titles basically have biology and engineering right in them. So, we sort of have an embodiment here of what the new field is and where it's going. So let me just start all the way at the very beginning of trying to just define the terms of synthetic biology. I think it's a term that gets thrown around a lot without people necessarily knowing exactly what it means. So I actually kind of want to hear from each of the 3 of you, just very, quickly, what is synthetic biology from your point of view? And Chris, maybe we can start with you.
Absolutely. So synthetic biology really refers to all of the practices involved in improving our ability to engineer biology. And that is encompassing of new ways to engineer biomolecules like proteins and mRNA, all the way down to full systems like thinking about the re-engineering of whole genomes. So everything that accelerates that process is part of the field.
Now, Douglas, you're coming at this from an electrical engineering point of view, so I'd actually be curious to hear, you have a slightly different perspective on that. Is synthetic biology, is it engineering the same way that electrical engineering is engineering?
Well, I hope it is. So I kind of drank the Kool-Aid early on. That for me to get into synthetic biology, I had to believe in the fact that synthetic biology was going to be engineering, that it was going to allow abstraction, it was going to allow modularity, that it was going to allow us to separate what something should do from how it's implemented physically. And so as an electrical engineer, those are all really important things in electrical engineering. And so just as what Chris said, to me, synthetic biology really is to be able to engineer things. using abstraction and modularity and rules. And if we can compose systems rationally, then ultimately people like myself can get in and build tools and techniques and algorithms to do that.
So I want to just drill down on that a little bit further. So you're saying that it's not, engineering is not a metaphor here. You're actually talking about being able to manipulate DNA. You're talking about being able to do with biology what you would do if you were designing a piece of software or an electrical circuit. Is that accurate?
Similar ideas. What changes are the rules and the requirements and the environment. Those are new parameters, new things we have to learn more about. But it's very much not a metaphor in the kind of research I do and the approach I take to it.
Christina, you were talking earlier about design, thinking of this sort of through the prism of design, as synthetic biology is a new tool for doing design work. Can you talk a little bit about that?
Sure. I think for many people who wind up doing engineering, they're motivated by design. They want to make and build things as opposed to just study sort of existing systems, right? And that's certainly how I came to synthetic biology, was the idea that, biology is something that we're exposed to every day, and we interact with it every day. But instead of simply studying existing systems, I was very much motivated by sort of the design and sort of building aspect of engineering.
So I have to admit, I don't know how many of you I'm speaking for here, but when I first came to this, to me it's a little bit hard to really wrap my head around this idea. As much as I know about the potentials of genetic engineering, understanding what is the advance beyond genetic engineering, what it is that you're doing here that kind of defines synthetic biology as having more control over the biological world and what you can do with it. Chris, you were talking earlier about almost like an assembly line metaphor. Can you talk a little bit about what does that mean? Can you actually build things almost in a mechanical way that way?
Well, I think one of the things that's changed very rapidly is that when I was a student, maybe a decade ago, we used Microsoft Word in order to design our systems. So we would literally take these snippets of DNA and try to put them together manually in order
you would actually just call up the program on the screen and say, as if you were editing an actual written document, like, oh, here's a sequence of DNA, here's a sequence of DNA, and actually just like cut and paste like that?
I mean literally using Microsoft Word, but instead of writing a paper, you would have your little file with all your DNA snippets that you would have memorized as to what their function was. And design was entirely encompassed by taking those little snippets of DNA and putting them together into a string. And there are all sorts of mistakes that you're going to make that are part of that. You are limited to what you had in your file of those pieces. And a big part of synthetic biology has been trying to accelerate the process by which you can assemble DNA, the number of parts that you have access to put in your design, and then a computational layer above that so that you can actually think about very complex, sophisticated systems that you might want to build and how you should approach that.
Right. So then, how do you take this idea, this thing you're looking at on your computer screen, and actually turn it into an object? Douglas, can you maybe sort of help me again, kind of walk me through the process of how you go from a concept to a thing that's an actual DNA string and something, and what do you do with it?
Well, yeah, so it's funny Chris mentions using Word. When I first got involved in synthetic biology, it was kind of through Chris, and I assumed it couldn't be that way, and it was. So that really, as a computer engineer, I said there's a lot of things wrong with this fundamentally. Hope we can improve that. So the process might be that imagine you have lots of different parts in a database that you think have a certain functionality. You hope they approximately are going to help you with some part of the central dogma, translating DNA to mRNA or becoming a protein. So once you've identified those parts, then ideally you have them somewhere physically in your lab, in a dish or in a freezer. So then you can start with a computer program, perhaps, that says, I'd like this generalized functionality. It might say, well, here's some suggested networks that might look like that. And then there's some parts that might implement those functionalities in your freezer. And if you know where they are in your freezer and your plate, then if you had automated robotic systems or microfluidics that knew where those things live physically, you could then build that large set of possible things that might work. If you have those possible set of things that might work, you build them, see if they do work. Those that work and those that don't work, we can start to learn from those experiments to influence the next round of assembly. And eventually, as we get more and more information, hopefully we'll make these predictions earlier, more efficiently, and more cheaply.
And so then, in terms of the end product, let's say from a design point of view, you're taking this information, you're turning it into, you're translating it into molecular reality, but then at the design end, if I want a plant that does something different, that makes a pharmaceutical, that produces food more efficiently. How do you actually then translate that into a design end goal? How do you get from when you're cutting and pasting in Microsoft Word or now with better tools to the actual the physical tangible thing that you want?
A lot of the programming that we do within synthetic biology, when we talk about programming a plant that's going to make a pharmaceutical or any organism that's going to do something different, is basically encoding your program within DNA, right? And that comes back to what was said previously. And so we might do engineering of molecules, engineering of networks of molecules that are going to interact, and a lot of times eventually then we're going to encode the production of those molecules, both what is being synthesized in a plant that's going to make those pharmaceuticals, and also when they're going to be synthesized and when they're going to be turned off. We'll encode those at the DNA level, and then that becomes a program that's essentially read by the cellular machinery. And the cell will then execute that program. And so that's a lot, and then the DNA will be inserted into the organisms or the cells.
It's a fairly, I mean, it's mind-boggling to me that these things are even possible, much less that there are tools to do it, and thinking about it in engineering terms. But again, as Juan alluded to earlier, the number of people who are doing it right now is still relatively small. The field is still relatively new, and even the potential of what it can do is relatively uncertain. All three of you have been involved not just in doing this, but really in thinking about how to educate the next generation and sort of how to help other people do what you're doing, maybe get them there more quickly or help them think more creatively. How do you do that? How do you break people out of a very sort of kind of a very sort of siloed academic approach of how people have been trained professionally, kind of traditionally? How do you train more people to do what you're doing right now, to bring these two sides together and to think about engineering really in very different terms than it has been in the past? Anybody want to jump in on that?
Well, I think one of the things that really struck me about Juan's introduction is that in 1994, I wanted to go into computer science, and my father told me there was no future with computers. So he encouraged me to go into chemical engineering, and that turned out to be a good toolbox, and a lot of the people in the field are from that area. But what you've seen is that each individual researcher comes from very different backgrounds, and I think that the time is just about right to really think about what's involved in educating a cadre of students where their focus is genetic engineering and building cells to address wide problems, not just in therapeutics, but also agriculture and chemicals and everything else.
Yeah, I'll just add a couple comments, which is to say that I actually think the field is much larger and growing than sort of the small representation that you see here. And in part, that's because of the educational outreach that's been done by the field. I mean, iGEM has really reached out and I think touched the imagination and excitement of a lot of young researchers, and those researchers are then going off to sort of grow the field. So that's the first thing I would say. And the second thing I'll say about education is, at Stanford, we've recently started a bioengineering department, and we thought a lot about education there and a lot about how to train sort of people who are going to be experts in engineering biology. And some of that is really thinking about sort of modern biotechnology and biological engineering and trying to break the mold of how we sort of currently teach it. So for example, there's a laboratory class that's taught where there's no genetic engineering that's done whatsoever. They actually outsource all of the synthesis. And so it really focuses on having the students design their system, right? So design becomes a big part of it, and then they outsource the DNA synthesis. So they're not doing any cloning or sort of trying to cut DNA and put it back together, but that's being completely outsourced. The DNA comes back in, and then they can test their system, right? And so really trying to introduce kind of the newer technologies and new ways of thinking about design, fabrication, and testing to the students at a young age to sort of train them for where the field is going.
Yeah, so Douglas, you made a comment that at first you couldn't believe that it really was that simple, that you really could think about biology the same way that you thought about engineering and computer science. I'm wondering how much of the challenge in this field is not just not just mastering the complexities of doing this very difficult thing, but also mastering the sort of transcending the intellectual barriers of realizing that you actually can cross over between these two different ways of thinking. That you actually can, I mean, we think very much of biology is sort of squishy and unpredictable, and it's people, it's things that the people are dealing with, in the lab or in vivo or in vitro, but it's not something that it's not something as controllable and as rigorous as circuitry design or software design, but you're saying that in a sense it is, or it could be.
I'm hopeful it could be. I'll be the first to admit, now that I've become a professor and have my own experimental space, that by no means is this a solved problem. It's very difficult. And so I don't want to be going on record saying that it's just electrical engineering, synthetic biology, one-to-one. There are many challenges that still need to be met. What's been the educational kind of challenge for me to make that work is that if I became a computer engineer largely because if a microprocessor didn't work or the design wasn't working, all I had to do to get to work was work harder. That meant stay in the lab longer, design more, test more. I found that not necessarily be the case with biology, that sometimes there are unknowns that don't work. And so what I think is important when I'm educating the engineers and computer scientists that work with me is how to keep moving in the face of failure. So when you're engineering and something just doesn't work, how can we learn from that failure? How can we archive that failure? How can we make that a data problem about learning from data that's associated with failure? So again, I don't, what's been nice to me is that while it's not simple by any means, that hopefully by taking some engineering discipline or engineering approach, things that might have been considered biological failures could be engineering successes. And so if we can try to categorize those and archive those, I think could be really powerful educationally as well.
Now, another thing that I think of, when I think of engineering, I think of patents. I think of, I think of sort of, there are very specific ways that you control information. I know that in genetic engineering, there have been a lot of issues about how you translate intellectual property and how you control that. Chris, is that an issue that's going to be coming up with synthetic biology? Is that going to make it an even more complicated problem?
Yeah, I think that's definitely a key issue. And as Doug mentioned, one of the real paradigms that's growing quickly is the design more and test more. And with a number of different facilities that are coming online around the world that are able to synthesize DNA very quickly, you suddenly have the capacity to print out a lot of variations of a design or of an enzyme, and then try to patent or control the entire set of information around that. So one of the key ideas that I think is going to be explored is what is the relationship between that huge capacity to print DNA information and then how that falls into the production as we think of it now.
Right. So I'm sitting here, as a journalist, I'm thinking, if you had a system where I could take a best-selling book, I could take, I don't know, the Da Vinci Code or Hunger Games or something, and run it through a computer program and say, you know what, I've just invented 1 million variations on this book. I'm going to patent all of them, and I'm going to sell all of them. I mean, that would, in the writing world, that would wreak havoc. But that's sort of analogous to what the problem that you're dealing with in synthetic biology, isn't it?
There are some analogies. So you could imagine trying to take a book like Moby Dick, and then the patent in that lingo would say, well, if it's 90% of this book, 80% of this book, 70% of this book, and so on. And so you'd be trying to cover this cloud of information around this one particular copyrighted work. And so that's fine if you're generating a single book at a time, and that's worked well. But if you have a facility that can print out hundreds of thousands or millions of sequences, then if you allow for the coverage of a cloud around each one of those sequences, you can very rapidly fill out a space around what is possible in biological design.
So when you see students coming up and expressing interest in this field, what do you tell them? How do you guide them into something that has such a huge potential and yet so many unknowns at this stage? What do you try to convey to them to get across the enthusiasm while also helping them understand the magnitude of the work that lies ahead? Christine?
I mean, I think when I speak with younger students or students that are interested in this field, I think you want to cultivate the excitement. You want to cultivate sort of the creativity and imagination, and you also want them to have sort of grounding in where the field currently is, what the shorter term and longer term objectives of the field are, and also be realistic in terms of how complex some of the problems are, right? And that there is this potential, but we need to think very deeply and carefully about really digging in and addressing some of these problems if we want to get to what the ultimate dream and vision is.
Right, you want to get to the concrete result and not just to the excitement of the field.
And to do that, it's important that they take their training very seriously, right? They need to have a very good foundation in sort of biological sciences, right? Sort of developmental biology, sort of the physical biology and models of cells, so that they can begin to think about how they would design within that space. So I think you want to motivate that type of training for the students.
Yeah, just along how do we motivate students? For me, in an electrical and computer engineering department, that's an interesting challenge, because I can talk to a 20-year-old, 21-year-old kid and say, come work with me in grad school, and they'll say, but I have this offer from Google, Microsoft, making 6 figures, et cetera, and in a field that's well established and we know will be successful. And for them to say, I'm going to do this 180 and take my skills and apply it to a new field, it can be a little daunting. But I think what really is attractive is that there's a lot of low-hanging fruit. You really have a chance to be a pioneer in this field. The kinds of things that we establish now, I believe, will set kind of the stage for the future. So a lot of students are excited by that aspect.
Well, certainly, I think if you look at what's happened in electrical engineering, in computer science, you look at Moore's law, the idea that you could apply Moore's law to biology. I mean, I think that if there's one thing that I think really kind of drove home to me the possibility of synthetic biology, it's that idea that what if you could, we don't really think of, biological advances are these very incremental things. I mean, traditionally it happened through breeding. More recently, it's happened through genetic engineering. But the idea that you could make progress in biology and that you could create new things in biology with the same kind of growth curve that you have with Moore's law in electronics, to me that's kind of mind-boggling. I don't know is that a useful metaphor from your point of view?
I think making biology accessible is useful. Like I think we all, every day we touch a computer, and even if you know it or not, you're programming it in some way. When you send an email and you push send, you're creating some program that does something, and you touch it. And I think that's what's been exciting to the students I've come across, is that they can interact with biology in a way they didn't think was possible.
You could be emailing biology. Cool. So here, I think let's take our first pause here for questions from the audience. Again, microphones on either side. If anybody wants to come up and ask a question to our panelists here. Do we have any interest? I know we have one person here watching the Twitter feed. So if there are any questions have come in through Twitter, not yet. Okay, we have a question right here. You can ask, and I'll repeat the question.
Yeah, biology, I think of as inputs, processing, and output. And so there's that big input question where there's all these expensive chemicals, tons of reagents. It's not like computers where you just plug electricity into it and you charge your battery. So how do you make it accessible when it's this very nebulous chemical cloud and still expensive? And how is this actually going to get into people's houses?
So I'll attempt to distill that. It's an interesting, somewhat subtle question. The inputs and outputs of biology are very different than the inputs and outputs in software engineering or in electrical engineering. Again, what I was saying before, biology is kind of squishy and messy, and so how do you actually make synthetic biology into something that is broadly accessible? When the DNA and all the other chemicals and the reagents and all those things that it takes to actually make this stuff are complicated, are expensive, and are not things that it's not just like a kid can do sitting at a computer at home. So, anybody want to tackle that?
Well, I think to start to address your question, one way to think of it, you're already halfway there in the way that you articulated it, in that just by dividing the system up into inputs and outputs and some sort of computation in the middle, you begin to think of the organization that's necessary. So, for example, an input that comes from a chemical signal creates a signal in the DNA, which can then be converted with circuits that function like electronic circuits but are encoded in the DNA, and then control the various biological responses as outputs. And once you've abstracted a system like that, you can actually look at each of those pieces in a very systematic way. So Doug and I, for example, work on the computational piece there, where in how the signals are integrated and control different responses, we're trying to make that look more and more literally like a programming language, where you could specify what you need for any particular application.
You have to understand the whole system before you can really do that effectively. You can do it in little bits and pieces. If you're trying to improve somebody's health or cure disease or...
Actually, let me pause and see quickly if there's another question from the audience, just because our time is so tight, but there will be time for follow-up afterward. Any other questions from the audience? There's...
Sure.
Yes, there we go.
I would just like to, maybe just for the sake of being provocative, take a contrary view in terms of the educational portion of what you talked about, but also takes a point of view from an engineering perspective, or at least consider engineering, is that the big successes come from teams of people who bring expertise from different areas to bear on the same problem. So one could argue that it might be better to train somebody in chemical engineering, somebody in electrical engineering, computers, somebody in biology, and then bring them together to solve problems, so that at the undergraduate level might be the place to train true experts in these areas that are going to impinge on the problem, and then maybe at the graduate level have things like systems biology programs or bioengineering programs. And again, I'm just saying this to be provocative, but it's something I think we have to think about. Do we want to have jack of all trades with masters of none, or masters that we bring together as teams to bear on the problem?
Okay, I'm going to just repeat it quickly so that we have it on camera. So the question is basically, are we getting ahead of ourselves here? Or, in trying to make students interdisciplinary too soon? Do you need to really train people in their individual specialties, and then you bring teams together, use kind of collective wisdom, rather than trying to, I guess, have several different worlds occupying, living together side by side in one person's head?
Yeah, so I would agree with the sentiment that you expressed, which is that it oftentimes takes interdisciplinary teams of researchers to really push on important breakthroughs, both in academic laboratories and in industry. I think, one point I would make is that I think when I think of a biological engineering major and curriculum, I don't think of it as a jack of all trades. I think of it as a specific discipline with its own foundational core that is distinct from chemical engineering, mechanical engineering, so that it brings its own set of expertise to the table when you talk about bringing those teams together. And I think it's a big question in the field in terms of what that foundational core should be for biological engineers, and it's not always executed correctly. But I think when we really sort of approach modern bioengineering departments, right, we really have to think about that and answer that question carefully. I will say that I've taught courses that do actually have interdisciplinary groups of students, and that we have done sort of team design projects, and we do try to get sort of mixtures of chemists and biologists and bioengineers and electrical engineers on those teams to try to think about design challenges.
And Chris, you wanted to weigh in on that?
Yeah, so I would say that it's not so much being a master of none. What we're arguing for is that there's a utility in being a master at engineering living cells, that as a discipline is something for which there's a demand out there, and for which really no single program currently trains people with that exclusive concept in mind. In chemical engineering, a little bit of the molecular background, but we were also working on rotary drum filters and so on, like that. In electrical engineering, you've got your distractions as well. So the question is engineering cells, living cells, a compelling enough need in our industry for us to be training students specifically to do that well?
And that's actually a great segue to our next panel. So thank you, three of you, very much. We're going to take a very quick break. Don't go anywhere. It's just a break on stage where our next three panelists join us. Thank you very much to the three of you. The first panel was kind of sort of establishing the lay of the land. For the second panel, we're ready to sort of get into more of the kind of the nitty-gritty of what you can do with synthetic biology. I think it's an exciting thing to be sitting here and thinking, this is a programmed biological system. It's a photosynthetic system. This is a programmed biological material that some code turned raw materials into this wood. I'm kind of a programmed biological system here that I think I'm working okay. You can be the judge of that. If you wonder what are the applications of what you can program with biology, the answer is basically the world around you. The real question is, do we have the tools to control that, and what can we do with that? And so we have our next panel can speak a bit to that. We have Virginia Ursin, who's the technology prospecting lead and science fellow at Monsanto. In the middle, you already got a chance to meet Jay Keasling, who's the head of SynBERC. He's also a professor of biochemical engineering here at Berkeley. And then Steve Evans, who is the fellow at Dow AgroSciences. So thank you to the three of you. And again, there's some interesting hybrid quality to this panel that we have private industry, we have academia, we have people who I think all three of you have really kind of moved back and forth, and they're very fluid between the two sides. I'd like to ask a little bit about basically synthetic. What is synthetic biology good for, based on your experience so far? And Jay, maybe you can start on this. I know Juan referred earlier to your work on malaria or on bioenergy. Maybe you can talk a little bit in specifics about what it is you're exploring, and we'll use that as our starting point.
Great. Well, thanks. It's a pleasure to be here. So one of the things that we've been doing with synthetic biology is engineering microbes to produce interesting chemicals. So drugs, you referred to an anti-malarial drug, artemisinin, that's coming out this year. It's going to be produced. They'll make about 100 million treatments a year. Biofuels, there's been a lot of work on those. Those are what I would call more of the immediate applications. You have some of the more long-term applications, engineering new materials, engineering replacements for all the other products that we get from petroleum. All these, you pointed to the wood. Well, it just so happens that wood's covered with a petroleum product, and the cushion on the seat, well, the entire thing is made of a petroleum product, actually. And we're going to be running short of those eventually, right? We might be able to replace some of those.
And then we have two people here who've been involved, on the agricultural side of things, or at least for companies that are involved in the agricultural side of things. I'm curious. Certainly, I think a common perception of American industry is that they're very focused on near-term payoff. And so I'm curious. First of all, if you're here because you see near-term payoff to synthetic biology, and if not, what is the interest in synthetic biology to a company like Monsanto?
So near-term for us I think is probably quite different than near-term in manufacturing or on the other industries, when the life cycle of our products are 7 to 10 years from proof of concept to commercialization. And that doesn't include the several years of discovery work to reach a proof of concept. So
we're not going to see synthetic biology products. You're not going to like go to the grocery store and see made with synthetic corn next year.
That's right. However, and in fact, I do believe that probably the earliest impact that synthetic biology will have on agriculture is going to be in the engineering of single-cell microbes. And so, as Juan said, we're a mosaic. We've got our human side, and then the microbial plants are the same. It's no different above and below ground, and we're learning more and more about how those communities, that rhizosphere and phylosphere, are supporting the health of the plant. A plant can't live without that, and I think the early potential could be in engineering those microbes to provide inputs. We all know agriculture is tremendously resource intensive. And so if we can use some of the early work on single cell engineering, I think we're a ways from multicellular organismal synthetic biology reaching marketplace, but I think that's where there could be tremendous near-term impact.
And Steve, what about from the Dow perspective? Is this do you see the same sort of thing, that this is not a 1 or 2 year cycle? This is a many year cycle of what it's going to take to see the payoff here?
Right. I'd like to answer it from that perspective, and then maybe expand it briefly to the Industrial Advisory Board that's part of SynBERC also. But so from our company's perspective, we compete in the same spaces, we work in the same technologies, and our time frames of 7 to 10 years are not that distinct. I would say that, as the first panel mentioned, though, we are engaged in the community that is coming out of synthetic biology is as employees. We're hiring people because they're bringing in alternative thought processes and concepts to help change the way early development might conceivably start. So new ideas, new hypotheses, I think, are important. But then if you step back, when you look at the industrial interaction with SynBERC, there are 25 companies that span the gamut, from actual software companies to large multinational companies in the areas of medicine or foods and flavors and things of that sort. So there's a spectrum, and the impact of synthetic biology to those different companies is on very different timescales. And so there are member companies that are directly impacting and being impacted by synthetic biology today, that are involved in this great consortium. So
I'm curious. So what are the near term, or who's being impacted today? Where are we going to feel this first?
Companies right now that are making software packages, that are making kits that are selling the DNA on a scale or in a magnitude that was simply not available just 2 to 3 years ago, are already important players in the way that companies are doing research. So the idea that you could get a very large number of complex genes synthesized and delivered in a FedEx package is something that was not practical just a decade ago. So synthesis providers, software companies, and then I think those are two immediate players right now. Then of course molecular biology has had a long history of being operated by kits, tools, technologies that read those kits and tools. And synthetic biology, in order to characterize many of these parts, needs more robust systems for capturing, integrating, and building on that data. And there are companies now that are involved in that interest.
Yeah, I guess we're just starting to get used to the idea of home DNA kits and things like that. Does that mean that we're in a few years we're going to have to get used to the idea of home synthetic biology kits, where you can put together sort of kind of assemble your own genetic sequences from modular bits? Is that where we're headed?
The kit might be just a software package that you order everything out and it gets delivered to you. So it wouldn't be like the standard chemistry kit, but might come back to you through the mail, ordered DNA, or even into a cell, right? We're going to get to the point where we can synthesize entire chromosomes. We've already shown you can do that on a small scale, but you could do it regularly even.
So a company might actually design a sequence, outsource it, and get back a package of, here's the genome built to your specifications. Now you can do the testing that you need to do with it.
That's right.
My mind is now officially blown. I hope I'm not the only one here. Since we have, I want to stick a little bit more with the agriculture question, since we have a couple people here specialized there, and since it's such a big area. The green revolution. That basically, the reason why all the forecasts that were made in the 60s and 70s of an incredible shortage of food globally, the big reason that didn't happen is because of nitrogen fertilizer and a few other basic but incredibly powerful advances that drastically increased the amount of food production worldwide. But there may be better ways to to do that, better ways to help plants get nitrogen, be more productive, and ultimately feed more people. Ginny, can you talk a little bit about that?
So as I told you earlier, in 1982, when I started graduate school, I wanted to work with Ray Valentine on making corn able to fix its own nitrogen, and that was. Well, I was 12 when I started, so 30 years ago, and we haven't done it yet. And it's possible that working the way we've done it, with basically within the biological constraints of the organisms we're working with, that it may not get us there. That it may take rethinking these pathways, refactoring, which is being done, and then coming back into these organisms and saying, here, this, try this on for size, rather than going in and working within the biological system that we've been working in. I think that incremental improvements probably aren't going to get us where we need to be in 10, 15, 20 years in terms of food security. Arable land is decreasing. We know that. With climate change, is our ability to use both conventional breeding and biotech even going to keep pace? We don't know. I mean, we're working on models. If we could have step change through this type, synthetic biology engineering, if we can move that into the question of agriculture, I think it is going to be another revolution. Absolutely.
Now, Jay, I want to circle back a little bit to the two things you mentioned as relatively near-term things that are closer to being used in the real world. Specifically, the malaria medicine, artemisinin. Can you talk a little bit about kind of where we are with that project and what it does?
Sure. So for those of you who don't know about the project, artemisinin is the world's best antimalarial drug. It comes from a plant. It's an ancient Chinese therapy. There are huge swings in price and availability. So to decrease the swings in availability and dampen the price swings, we took the genes out of the plant and put them into a yeast. And so the yeast spits out the antimalarial drug, or a precursor. And it's a process that's just like brewing beer. That's been scaled up now, and Sanofi's produced 35 tons of the medication, which is enough for about 70 million people. And that's going to launch April 11th is our launch party. And drugs will start going out this year with the artemisinin in them.
So we're 2 weeks away, roughly, from launch here?
Yeah, we're about 2 weeks away from launch.
Okay, good timing.
And then Sanofi will produce on the order of 100 million treatments on an annual basis, which will cover somewhere around a third to a quarter of the need in the world. There's roughly 500 million cases annually, kind of at the upper limit, 300 to 500 million cases. So it'll cover a good amount of the need.
And then the other one, the biofuels, yes, which I think we went past a little bit quickly. I want to understand what's the work there, because certainly, I mean, from the media side, we hear stories all the time about the incredible potentials of biofuels, and then all the hurdles to actually making these be commercially and economically useful.
Right. So the potential is that we have 1 billion tons of biomass that goes unutilized in the U.S. On an annual basis, and if we could turn that into fuel, we could roughly produce a third of the need in the U.S. And there are a lot of innovative companies that have come up with new organisms that will produce advanced fuels, so not ethanol. Ethanol is for drinking, not for driving, but actual fuels that you can use in your automobile. In your plane.
Yeah, I can actually hear the Twitter characters forming as you said that.
The challenge, though, is affordability. How do you make a fuel that's going to compete with a petroleum-based fuel, with a fuel that's cheaper than the water in that bottle? That's a huge challenge. And so what you see with many of these companies, even though they have great promise, is that in order to stay alive, they're going to be making other chemicals, replacements for all the other petroleum-derived chemicals, because those happen to have bigger margins, and therefore they have a better chance of staying alive, or at least getting to profitability. So I think it has huge potential, and some of these can actually significantly reduce our carbon footprint by, say, 80%.
We're going to break for another set of questions in just a second, but before I do, since all three of you think are thinking in terms of near and midterm commercial applications, where are other places where we might see synthetic biology in the relatively near future? Where do you see the most likely commercial or applications? I think beyond the things that we've talked about, is there anything else that comes to mind?
I think enzymes that are currently used in manufacturing has a tremendous potential. So, as we talked earlier about the inputs, the magic happens in outputs. So the potential of synthetic biology to give tremendous new diversity in the chemical reactions that can take a raw product, an input, into something value add out the other end, I think it is happening now, and I think there's going to be tremendous potential there.
Okay. Any other thoughts before we break?
I'm hoping for synthetic biology applied to hair growth in the very near term.
Amazingly, I am too. Okay, let's open it up to questions from the audience again. If you can make your way to the microphones, great. If not, raise your hand, and I'll do my best to work on it. Because of the lighting here, it is a little bit hard to see what's going on. But yes, I see a question back there.
Can any of you speak to the potential for creating synthetic genomes and applying them as probiotics in the environment and for agricultural uses?
The question is okay.
That's actually what I was alluding to earlier in talking about, first of all, understanding the associative, the biomes that are associated with plants, and then so probiotics is another, there's endophytes as probiotics, and understanding what they do and then engineering that to increase their impact on the health or protection against disease, I think is probably going to be one of the nearer term impacts of synthetic biology on agriculture.
I think one allied area also, in looking at spaces, say, around bee colony health, the symbiotic organisms that are involved in bee health also might be a potential area in which some sort of adaptation, modification of the organisms as a probiotic concept might be useful.
And even synthetic organisms that we could have in our yogurt. So you could inoculate your gut with a culture that might make you resistant to malaria and to other diseases.
Do we have any other questions here? We have time for one more quick one before we do another quick switch. Is anybody? Well, so if not, so one thing I wanted to just follow up very quickly on that idea that what's interesting about synthetic biology is that it's a bridge between so many different sets of ideas. Things that we typically put in these separate silos of, a probiotic is this sort of, is this kind of holistic organic thing. Synthetic biology is this sort of genetic engineering type thing that is, there are these things that we create these kind of categories in our heads of what's synthetic and what's natural, and really there's a whole blurring of that so much of our natural world is bred and hybridized and kind of adapted to our needs, and the flip side that you're talking about using synthetic biology to actually, in some cases, say, reduce the chemical footprint of agriculture, or reduce the environmental impact of creating energy. I'm just curious if how do you talk to people about that? How do you get them to think about to rethink that difference between what is synthetic and what's natural, or sort of what's healthy and what's unhealthy?
That's a good question for Monsanto.
Yes. Since Monsanto, my impression is that Monsanto has occasionally faced that question.
No, I actually think I think certainly the diversity of life as it exists. To say something is natural because it exists out there is fine, but that diversity is beyond anything that we can comprehend. And so is something non-natural when it falls outside of that spectrum? Maybe, but we don't even know the boundaries of the spectrum. I mean, if you take things like the microbes in your gut, I mean, it's communities that I think it's hard to even dissect into. These are the organisms that all this DNA that's in there belong to. DNA is moving tremendously fluidly between colonies of organisms. So, you know, I think it's a hard question because I think we don't understand where the boundaries of natural are. And even if we could, it would be changed. An hour from now, it's going to be different. Mm-hmm. And so I think, you know, working within the way in which we understand how to show something is not harmful is essential. But to try and put things into a category of natural versus unnatural is somewhat artificial because we have no way to categorize natural. We have no way to put boundaries around that.
And in defense of all of this area, I am from a farm in Nebraska, and when I grew up as a kid, my father was spraying all these pesticides on the crops to get rid of rootworm and any number of pests that would attack the crops. And I'm actually surprised that he's alive and that I'm alive because of all that. And it changed dramatically when we started getting Roundup-ready corn and some of the other engineered traits into the corn. The environment cleaned up tremendously. And started using less water. It's amazing what the transformation has been. So from an environmental perspective, it's been a huge improvement.
I think it's important to understand what we're talking about here, synthetic biology. It's a continuum of this revolution in biology that's happened in the last 20 to 30 years. There really isn't a line. So on this side of it, it's molecular biology, genetic engineering, this side of it, synthetic biology. This is expanding the toolbox to make outcomes that we couldn't imagine even 5 years ago.
Okay, we're gonna have to pause here, but obviously this is not the end of the conversation. This is the middle of the conversation. So thank you very much to our second panel. And we're back. You may not even know you went away, but we're back. So this is the third and final portion of our conversation, and I'm hoping kind of continuing the arc of where we're going, looking at sort of different scales of the possibilities of synthetic biology. We're going to try to take the sort of the biggest and longest range view. With our third panel, and we're very fortunate to have George Church, who's professor of genetics at Harvard Medical School. Laurie Zoloth, who is professor of medical humanities, bioethics, and religious studies at Northeastern. So you've pretty much got I'm sorry, Northwestern. I'm so busy trying to get all of your subfields right that I got your institution wrong. And then Drew Endy, assistant professor of bioengineering at Stanford. So I think I want to pull back a little bit further even, and we're looking at what are the long range sort of hopes and goals of what synthetic biology can do, and even and sort of what we understand about what it's capable of doing. And George, you've talked a lot about this. You've written a lot about this recently. I'd like to sort of get a little bit of your perspective, sort of something of your vision for the field.
Well, sure. We've already talked about green chemistry and pharmaceuticals, but there's smart materials. You knocked on wood, but in principle, every material could be the cost of something highly complex biomaterial like wood, on the order of less than $1 a kilogram. Smart materials could extend all the way out to molecular and three-dimensional computing, data storage.
Grow a computer?
You would manufacture it with biological principles, including growth of various sorts. You could developmental systems, human systems, gene therapy. Already, we're not growing hair, but we are definitely fixing individual individual T cells that can be fixed so that they become HIV resistant by gene therapy. This is already in phase 2 clinical trials. And then, of course, that same kind of technology that results in gene therapy that's working can be applied to habitat maintenance via de-extinction or preservation of endangered species, and brain activity map, optogenetics, and so forth is showing some incredible possibilities for input from the synthetic biology community.
It's a pretty breathless list. I'm going to want to come back to some of that, but I also want to let their other two panelists kind of weigh in on this. Drew, can you talk a little bit about your vision? You really kind of sort of helped establish the idea of kind of modular building for synthetic biology. And maybe can you talk a little bit about kind of where you see that going, of actually helping people have a toolkit for synthetic biology?
The ideas predate me, and they go way back in engineering, right? The aqueduct in Spain, that the Romans make with the standard rocks, the modular rocks. So can you port those ideas into the molecular world? I think a decade ago it wasn't obvious for some types of molecules, but 10 years in large part through the fundamental investments of SynBERC, we've shown that it's not impossible, right? That you can begin to make things more composable, more reliable in a forward engineering context, so that a would-be engineer biology, if they immigrate from CS or electrical engineering, has a chance to compile down a DNA code, and it works. It's a shocking experience to go through that transition. I would just simply relate, in the last year, I've had the first experience with people on my teams designing systems, and their first DNA designs work, as opposed to 700 attempts. And what comes from that, I don't know, other than to imagine that a lot of it is going to be boring. That we're simply going to see a promulgation of some of these fundamental concepts, and folks will be upskilled in terms of what they can make with biology as a partner. I guess my vision...
I'm sorry, meaning that you'll know synthetic biology is happening not because something totally dramatically different has happened, but because the things that we were doing before are happening faster and more efficiently.
Well, but as you get immigration, and I think Doug Densmore said it pretty well, you make biology an accessible technology. It's a very compelling set of material, right? It's living stuff, it replicates, it's taken over the planet, it's a nanotechnology that works, just to paraphrase things you hear from others. And you can kind of code it, right? You can get better at coding it. That's very attractive. So I think there is something to getting this material in a programmable context into more people's hands. I think George and Jay both made some good points. In Menlo Park, where I live, we have a surplus manufacturing capacity. It's called the yards and gardens. We grow grass and pine cones and stuff like that. If you go to the Environmental Commission and pull the records, it's 16 million pounds a year of manufacturing, state-of-the-art nanotechnology. The input stream is not electricity, it's atmospheric carbon and photons. And we just throw it out with a little bit of composting. And so I guess what I would like to imagine as a longer-term sort of encompassing vision is that humanity figures out how to reinvent the manufacturing of the things we need, so that we can do it in partnership with nature, not to replace nature, but to dance better with it in sustaining what it means to be a flourishing human civilization.
So that's actually a nice segue to the next thing I want to talk about. So Laurie, humans have always been reshaping the environment around them. I mean, certainly almost as far back as you can see in the fossil record.
That's what we do.
Yeah, it's all we do, and it's not just humans. I mean, it's kind of the nature of biology is to consume and shape the environment around you. But I mean, at the same time, this is a sort of a transformative technology. This is a level of capability we haven't had before. How do you decide where you want to go, and how do you make sure? I think there tend to be a lot of somewhat circular political discussions of like, oh, is this a good technology? Is this a bad technology? Is this going to be used for good or evil? And they don't really take you very far. But at the same time, there are some very practical things we need to decide of, what should patent law look like, and what kinds of regulations should there be, and where should federal funding be applied? Where should universities be focusing? Who makes those decisions, and how do we make those decisions?
Well, let me go back a little, because I liked the question about what's our long-term goal.
Okay, sure, okay? Absolutely.
I too want to talk about the long game.
Okay.
So the long game for me would involve something like a world that's more just. A world that takes into account the fact that revolution, even a revolutionary potential, might be a disaster for some people. That what makes this interesting science, really interesting science, is that it could also fail. It might not fail. It might not succeed. It might not be so wonderful. And that part of the reason it's science and not, a faith claim is that it's an experiment. And that's what makes it extremely interesting. Some of what we hear is, I believe, possible, but I just believe it, off to see. Mm-hmm. And some of it isn't, and you'll have to learn it. Like the corn that should be able to be fixed with have fixed nitrogen in it and grow wonderfully there didn't work, or it's really hard, or many of the technologies that you thought about just are very hard. They're complicated. Biology is much messier than machines, and it's stochastic. And that's what makes it interesting. It doesn't make it impossible. It doesn't even make it wrong to try. And it's exactly to try to create a transparent and a just framework for allowing the science and the experimentation to flourish that's worth it, to take a look at this for fora such as this, for government regulators, for NGOs, for lots of people to look at the science and say, well, how do we make it both good and sustained and free and also just? So the work that attracted me was, of course, the work about malaria, right? Jay Keasling brings me into his lab years ago and says, I think I could cure malaria here. Mm-hmm. And that's interesting because not that many people cared about curing malaria. They actually cared more about hair. So,
okay,
there
fine.
Is,
you know what? If I lose my hair, but malaria is cured, I choose
totally
malaria.
So that long-term thrust, the understanding that the world is a broken place and that our responsibility and our obligation is for tikkun olam, for fixing a broken world, it's that's interesting, I believe, about synthetic biology. And that commitment, which has always been a part of it from the very beginning, needs to be equally nurtured by these processes that you speak of.
So let me follow up on that. One of the things that George mentioned was conservation biology. I think Juan talked about it in terms of de-extinction. Now, obviously, when you're talking about the total commercial potential of synthetic biology, being able to bring back an extinct species, that's not one of the big applications. And yet, in terms of as an intellectual application of what we could do to actually kind of undo part of our footprint on the planet, it's a very interesting and very provocative topic. Mm-hmm. Is that a real prospect of bringing back extinct species, and how would you go about doing it?
I think the important thing is that this isn't necessarily doing it for curiosity or even because we owe it to our past ancestors. It could be that we need it for habitat preservation today, but this needs to be very clearly articulated and questioned, cross-examined. For example, an argument has been made since the mid-90s that the permafrost, which has more carbon than all the rainforests put together, is at risk just by simple thawing, and that a large herbivore like a mammoth could maintain it for another few decades against global warming. If that's true, then that's is at least as big an imperative as returning something that we killed off 3,000 years ago. How would we do it? I mean, the techniques are similar to the ones we're developing for gene therapy, combined with our newfound ability to read DNA even when it's broken up in little pieces in museum samples or in the permafrost.
So, Drew, I'm curious. Do you see? I mean, these terms of of justice or maybe of sort of effective caretaking. How does that play into the kinds of things that you're talking about of sort of expanding the possibilities of what people are able to do and build?
It's a good question I don't know the answer to. Mm-hmm. As a surrogate, I think about Joy's Law. After Bill Joy, previously of Cal, then Sun, most of the smartest people work for somebody else. And then implicit is, so then what's your strategy for dealing with that? And I think what's interesting about biology and the future of biotechnology is that Joy's Law ramifies in almost every way I can sort of play around with it. We operate today still largely in a regime of ignorance with respect to the details of the living world. Most of it remains to be discovered, and by definition, most of those discoveries will be made by people, not us. So as a would-be engineer of biology, I need to think about how I'm going to scale my interfaces to the rest of the research community. In a world that's a changing world, we think we're familiar, for example, with the internet and its separation and reconnection of space and location and information, but it's still very, very new. just we're still trying to, is Twitter going to work for this event? That's kind of trivial in a context where the rest of biology is to be discovered. Most of the smartest genetic engineers are not going to work for me. Most of the needs and opportunities that could be addressed with biomanufacturing, those will exist somewhere else. I think that's a unique opportunity in synthetic biology because biology is the ultimate distributed manufacturing platform. Samsung, as a consumer electronics company, becomes a lot more interesting when you think of the bread machines they might sell as biomanufacturing platforms, where your loaf of bread is a pharmaceutical company, right, making the drugs you need in a distributed asynchronous fashion. Obviously, most of the bioterrorists, the best bioterrorists, are going to work for somebody else, right, and so on and so forth. So what's my strategy for operating in a world where the things I need and the things I want to do and the problems I might cause, directly or indirectly, are all going to involve many other people? And that's where I spend a lot of time working through struggles to think about, is this the right thing to be making easier so people become more capable in this dimension, or do we back off here? And nobody really comes up with good responses to that, so we're always just sort of struggling along, I would say.
One of the things that it's interesting to think about is when you asked about regulation, because the products are so by design mutable, right, and the methodology is so swiftly changing, I'm more interested in thinking about what this technology makes of the women and men who are doing it, what sorts of interior moral choices they need to be making, and how you create scientists who aren't only good at all these different technical skills, but are very good at asking and thinking seriously about ethical questions, about moral questions, and coming to terms with the ramifications of their work. That's something that's an ongoing process. You raised the issue of nitrogen, which one of my favorite stories. So in 1898, the world was in free fall, catastrophe mode, that people were going to die because all the nitrogen in the world came from a few islands off the coast of Peru and Chile. And there was a war there. They had to be trucked in ships all around the globe. And there wasn't enough. They were running out. They came to the end. And so the British Academy of Science raised the kind of alarm that you hear in global warming, said about it exactly what George Church just said, is maybe this can be solved by the lab. Maybe the lab is the salvation of humans. And it turned out that was the case, that the smartest people were synthetic chemists. And they pulled nitrogen from the air, one of them, Fritz Haber in particular, setting up the Haber-Bosch process, pulled nitrogen from the air and made it possible to completely transform nitrogen into a product that could be made and then applied to plant growth all over the world. Now, it had tremendously great application. It saved millions, perhaps billions of lives, because people could get enough food, and ordinary farmers could get food then. And in fact, the next problem they had to work on was what to do with all the rats that were now drawn to these huge storage houses. And he began working on rat poison. But, right, which turns out to be an interesting part too, because the rat poison that he developed was called Zyklon B. And Haber himself was a Jew who had converted to Christianity. When in World War I, he and the other leading French chemists of the world got turned to by the various governments to say, you're the best chemists in the world, stop the carnage of World War I, make a chemical product. And they both did. And then they used it against each other. Gignard and Haber, who had both won the Nobel Prizes, ended up making World War I the chemists' war. By World War II, Haber had come to decide that he could no longer serve the government in that way. No one would shake his hand, for one thing. And he was asked to fire all the Jews in his lab and use the Zyklon B as a weapon of war, and at that time he refused, right? So if you follow the story of nitrogen and of one chemist developing synthetic nitrogen, you see all the same joys and temptations and capacities that these guys are faced with here. You could change the world, and you have a powerful technology. And you need more than external regulations. You also need a deep moral process that allows you to see and name what you think is going to be important, and how the end game, what you want the world to look like, and what's the goal of your science, what's the telos of your science. And so I tell that story because it's so fascinating in light of our current crises in climate and ecology and environment, which frankly, without this work, I don't think we have such great answers for.
Thank you so much for that. Let me, since we're just about at our wrap up, I'd like to get from George and Drew as well, sort of, if I could sort of kind of put you on the spot and say, what would you like people at this conference, people who are going to be reading about this or watching this on the internet later, what is the most important message for them to come away with in terms of understanding what synthetic biology is and what it could do? I realize it's one of those big global questions, but obviously it's something you think about all the time. George, what is the most important message for people to come away with here?
Well, I think that the transition, the transition that we've made from genetic engineering to synthetic biology is a transition to a full-fledged engineering field, which includes safety engineering. And I think what Lori is talking about, what we're all talking about, is we have an obligation to do it right. And because our environment is changing, our world is changing, the decision to do nothing is a gigantic risk. The decision to do a particular new thing is a risk. We have to get better at risk assessment. We have to get better at safety engineering. I think this is an incredibly important opportunity and mandate that we have.
Drew, you want to have the final word here?
If you look at the room we're in, there's some biology in the room, but it's very crudely engineered, whereas most of the other materials are quite sophisticated in their engineering, from the Stone Age surface that we're sitting upon to the electronics recording and broadcasting the event. And I presume that's true for most of the audience here in the room and on the internet. So what does the world look like, and what do we want the world to look like as over the next decades we get actually really good at engineering biology? We make living matter programmable. My hope would be that people recognize that incremental advance, and it'll seem, it's funny, like these words, revolution, incremental, whatnot. The neat thing about engineering is in the heart of engineering, you've got this design, build, test cycle. And you go around it a little bit faster, you go around it a little bit smarter, you go around it more times in parallel. And incremental improvements in going around that cycle, sustained over time, do deliver geometric increases in your capacity to make stuff. And my hope would be that people don't see that as a passive process, that because we're talking about living matter, we're talking about biology, that folks all around the world participate and become citizens with and of biology as we become better partners to engineer it.
Thank you so much. Do we have a moment for one last? Okay, so we actually have just a little bit of time for one or two final questions from the audience. Let's see. I see one back here.
Thanks. Ben Chorney from UC Berkeley. So I'm wondering, given what Professor Endy said about how much we don't know about biology, how much do we need to know about a system before we can engineer it safely? And how do we know how much we need to know?
Their operational definitions, those are pretty useful, right? The physicists are still happy to study gravity, but we have a suspension bridge over there, over the Golden Gate. And so very pragmatically, you can observe what happens when you try things and learn from that experience. I think there's a good comment made in the previous panel that synthetic biology, although sometimes it's thought of as being ab initio, just new, it really in many respects is an outgrowth. And biosafety as a particular topic is a place where synthetic biology depends upon past 40 years of experience with recombinant DNA, genetic engineering, and biosafety based from that. And if you look into the details of those frameworks, the approach taken typically is to try and contain things within physical settings, if not biological settings. I think one of the interesting puzzles and challenges that SynBERC spends a lot of time on is to think about what happens as the applications of biotechnology expand well beyond what you've seen in laboratory settings or in agricultural production settings, and how do we assess safety, how do we build in for safety, and just up the game, so to speak.
Let's see. We have one more question. I think we can sneak one last one in. Do we have If we don't, anything off the Twitterverse? Okay. All right. I think that's a fabulous place to end. Thank you for the three of you. Thank you for this whole panel. To SynBERC for hosting, and thank you all for being very focused through covering a lot of material. So.